Hey everyone, we're going to go over the lab design that you guys picked up. If at this point you have not read through the background information, please pause the video and read this information first. And then when you are ready and you have read it all, go ahead and hit play and resume following along with us. So the first thing that you need to do when it asks to find the two hypotheses in the research and underline them, there's going to be two that we underline. Here is the first one, where we look at how does the color affect the number of responses and mates that these butterflies get. So that's hypothesis number one. And then the second hypothesis is right after that. What happens if we keep the same colors, but we have a different pattern? So those are the two hypotheses that we're going to be looking at in this experiment. I do need to apologize. I am a little congested thanks to allergies, so you'll hear some sniffling on the film. The next thing that we're going to do is we're going to go through and look at the different parts of your we're going to be looking at the different parts of the lab design video. So we always look for a control. And in this case, model A is going to be our control, because model A has what we call the normal conditions, both in coloration for hypothesis 1 and the normal or local pattern for hypothesis 2. So then how are they changing? How are they manipulating that independent variable? Well, in model B, they're going to have the same pattern but different colors. So that's how they're changing that independent variable for hypothesis 1. And then for hypothesis 2, they're going to have a different pattern but the same colors. So in both of these cases, they're going to be using so that's how they're manipulating the independent variable. And in both these cases, they're going to be using two factors that remain the same. In hypothesis 1, it's the same pattern, using the same printed butterflies, paper and printer. And in hypothesis 2, it's the same colors with the red and yellow, and the same paper and printer that the other model is made out of. So they're going to measure their dependent variable by watching and counting how many times each of those butterflies get approached, each of those models get approached. And then that's how they're going to measure the dependent variable. And then the last thing you always look for is multiple trials. They're using 51 butterflies, not just one or two or three, but they're using multiple butterflies to test out their hypothesis. Down at the bottom, you're given a data table and asked to interpret it. I'm going to give you a minute to look over this. So you were given a piece of graph paper that we're going to glue to the top of page 27. Go ahead and pause the video now and try drawing your bar graphs and your standard error of the mean. Keep in mind that this standard error of the mean has already been multiplied by 2. We're already doing 2 standard error of the means. So go ahead, press pause on the video, add this to your notebook at the top of page 27, and then when you are ready, go ahead and hit play again to see the answers. All right, so this is what your chart should end up looking like. You'll see that we have models A, B, and C based on what the scientists were testing. We've also gone in and drawn the standard error of the means to two standard error of the mean bars. And we're looking at the different populations. So in this lab for hypothesis 1, we're going to compare A to B. And so we have two different conclusions. We have two different claims that we were making. So based on the data between models A and B for hypothesis 1, more butterflies approach the brightly colored model than the black and white. Because the SEM bars do not overlap, there is a statistically significant difference between models A and B. And then for hypothesis 2, we're going to compare A to C only. Based on the data between models A and C, butterflies did not show a preference for either pattern because the SEM bars did not because the SEM bars did overlap, there is not a statistically significant difference between populations A and C. Hope this helps.